else, and they're setting up. I have to sit down here because I have a bad back and hip, so it's also to see the slides and everything else. So I hope everybody can hear me and everything else, and they'll be setting this up. Again, this conference, I was here, I guess the first conference many, many years ago, invited by the House family, got to meet Hans and Arnold, who are, him and I are about the same age. Maybe I'm a little older, maybe I'm a little younger, I don't know. Oh yeah, we're the same then. And of course I met Nina then, and met everybody, and met the House family, and I think it's this one here, okay? And since then, we've been working on Pompe's disease and the, molecular biology of that, and then that sort of has, thank you very much, that has sort of gone the wayside along the way, and I'm a simple person and doing simple experiments. I'm not sophisticated doing that, and if I need something sophisticated, I call the grand poobah, Nina, and I always get her input and everything else, because it's so wonderful. Currently, we thank Tiffany for inviting us to present our simple studies and everything else, and Randall and Marilyn House should be really complimented, because they're the ones that started all of this, getting the scientists that are in, our scientists, all of us that were in our silos many years ago and not sharing everything, and getting it to the patients, and basically kicking us in the butt and getting us to communicate with each other and say hello to each other. Besides, of course, the internet wasn't around then, and getting us to share data and really to do it for the patients. And they deserve the credit. We also like to thank, really, and they're so modest. They won't accept it, but they're so modest. Now, let me see. We also like to thank, Marsha for help in the background, making Tiffany's life easier. Currently, because of funding and everything else, and basically Myozyme has put a lot of getting money for research, basic research, very difficult. Now I'm currently with a very small company called JME Group, which is only 3 of us and a few volunteer, part-time technicians. We're in New Jersey. We have a lab in New Jersey, and we're basically to develop and commercialize cost-effective enzyme replacement therapy for metabolic and lysosomal storage disease via oral administration, Oral-ERT, with plant-derived recombinant enzymes, and also a non-GMO plant products for improvement of human and animal health. The first enzyme, of course, that we were looking at, it's been working on it forever, is acid maltase, and it's a recombinant enzyme produced in tobacco seeds, which it's called TOB rhGAA, just to give it a name and everything else, something a little simpler for the treatment of AMD, or acid maltase deficiency. I'm not going to mention anything because I'm not an expert on it, and all the clinicians and the patients are really in the trenches with Myozyme, and they're working it out and finding all the limitations and the positive signs and the negative signs of Myozyme. So, I'm not going to mention that, and of course you guys are doing a great job and really moving it forward and trying, and eventually, as was brought up yesterday, there will be a combinational therapy that not even personalized, but I think we were saying that it'll be a whole battery of things thrown together in a however it's administered, enzyme replacement, gene therapy, chaperonins, all kinds of things thrown together for the patients, and hopefully that will be the cure and everything else. Although Myozyme has been really a wonderful first step in treating AMD, it has revealed a lot of other subtle aspects that must be dealt with for successful treatment. ERT usually begins before, when the patients are symptomatic and secondary problems are already present. On the left side of the slide here in red, this is some issues that are with Myozyme that I keep simply put, and there's a lot more of them, basically, some of the problems with Myozyme is there's an inefficient uptake. There's a transient nature to the administration, either 2 weeks, 1 week, however, and there's changes in that. Glycogen reduction, which is very important, is variable. There's autophagic, mitophagy, and lipofuscin accumulation that, Nina is really in the forefront of describing all of that and trying to work that out. There's a potential immune response depending on the patient and whether they're CRIM negative or CRIM positive. There's an extremely high cost, which everybody knows about. The effective residual level is something that, what is their effective residual level for treatment and everything else that you need to accomplish. And then the Myozyme, I think it needs, since I've never handled the bottle of it, I assume it needs refrigeration, and it clearly needs a clinic visit, or you have to come see a physician to be administered and everything else. On the right side is our, in blue here, is our oral enzyme replacement therapy with the tobacco TOB rhGAA. And I'm sort of putting this up front and with the slides that will hopefully chronologically how we sort of got to this point, and some of the benefits that we think by this product and the administration. And I shouldn't put efficient. There's clearly uptake in appropriate tissues. It'll be administered and eaten daily in either a whole seeds or a ground up seeds, in either a capsule or put on food, or however you want to drink it. That is main to be seen. We haven't done the autophagy and lipofuscin stuff, but that's going to be ongoing. We have all the tissues put aside. Because it's given orally, there's a minimal immune response because of immune tolerance and everything else. And then projected cost, which is for plant-made pharmaceuticals, which is called PMPs. And the projected cost is in the neighborhood where we think is between 1% and 10% of Myozyme, and that depends on whether you want to have to purify it, and we're not going down the route, at least at the moment, for purification, so it probably is in the 1% range of Myozyme. That's where it'll be. Because it's administered daily, they have an increased residual level of enzyme, whatever that is, and depending on the amount that is given. And then it's extremely stable at room temperature. And I'll show you some data for that for many years, and actually, at least, well, right now we're going on 15 years that the seeds are stable and the enzyme is perfectly happy being in the seed. And it requires no clinic visit because we can just give the patient this material, and they can carry it around with them and take it on a daily basis. So how did we sort of come down to this road and everything else? Due to the high cost of lifelong ERT, which is $250,000 to $500,000 per year for an adult patient, depending on their weight and everything else. More novel, affordable approaches for ERT what's really needed and everything else. Transgenic plants, seeds, cultured plant cells, are potentially one of the most economic systems for large-scale production of recombinant enzymes for pharmaceutical uses, and that's clearly biomass. You can, Myozyme is made in their reactors, and it's secreted, and it's got to be grown and maintained. Plants, you can basically grow as much as you want of it. There's unlimited resource, and it depends on the amount that's in the plants. Over 1/3 of the pharmaceutical proteins are glycoproteins, and even minor differences in N-glycan structures can change the distribution, activity, stability of the recombinant proteins. Recombinant seeds, and in particular tobacco seeds, offer a high recombinant protein yields, long-term stability, and the metabolic machinery more compatible with mammalian glycosylation, phosphorylation, cleavage, processing, critical for function of proteins, etc. On the next slide here, in 2013 we published, and Arnold, please tell me if I'm going too long, so I'll just stop. In 2013 we published this paper on the development of a transgenic tobacco plant expressing human GAA in seeds and everything else, and with all the authors there and everything else. We would have published it earlier, but however, there was patenting going on at NYU, and you can't publish something until you patent it, blah, blah, blah. That sort of is still ongoing with the patenting process. Previously, and being being simple, and when Mr. House visited our lab and everything else, we had expressed the desire, we wanted to express the human GAC DNA in chloroplast calyces, which is basically a clump of growing plant cells, which you can differentiate into a plant, and leaves of transgenic tobacco plants, but the enzyme was not active in these other organelles. Tell me when, okay, to stop.
So what's the data?
Yes, we're going to get to the data, but we originally envisioned that the patients would eat a tomato plant or have a tomato juice every morning. They'd receive their enzyme replacement therapy, but that didn't seem to be a viable system. So that didn't work out and everything else, and then our goal was to accumulate the recombinant GAA and mature seeds in a high quantity, and to stably maintain the enzyme, which was a promoter for soybean beta-congluten, for a seed protein. We made this construct with the human seed DNA. We replaced the signal sequence of the soybean with the soybean, and replaced the GAA signal sequence for proper processing and translocation. On this slide here, I'm not going to go into, this is basically the construct that we used, which contained all the important elements and everything else to get it into the plants. The plants were basically, they were transfected by triparental mating with Agrobacterium tumefaciens bacterium. 40 independent plants were selected by kanamycin resistance. By molecular analysis, 87% of the plants had 2 minutes. Okay, so we'll get to it. 87% had were transgenic plants. The GA activity and everything else, and Western analysis proved that it was in there. In this first part here, we show that the extract from it was completely active, and this was in the paper. It could be taken up by fibroblasts from patients, white blood cells from patients. It could be purified. We did a very simple experiment with Nina's mice, and we showed increase in enzyme activity in tissues, et cetera. On the bottom there, on the left side is the plants themselves and the flowers. On the right side, you can see how small these seeds are. They're very tiny seeds. They're like pepper. We received, so to keep this going and everything, we received an NIH SBIR grant, and we'll just now show you in a draft of the manuscript. We'll just quickly go through our data that sort of hopefully backs up our tobacco seed recombinant human GAA. This is with early experiments. We didn't know exactly whether it would work and everything else, and we gave it to the mice and everything else, and we gave it orally, and we fed a couple different doses of it, which are seen in the middle, starting from and moving on all the way up to day 203. The top of the graph shows them the wild type mice, and the bottom is the mock-treated knockout mice, and you can see that the enzyme activity has clearly increased with in vertical hang time measurements, and it was about 25 to 30% of the wild type animals, and at day 62. The interesting thing at this point was that the lower dose actually caught up to the higher dose of the enzyme and everything else. So we were very happy, and this sort of got us started that maybe we could give it to them orally. They were given 3 times a week, giving it to the mice, just squirting into their mouth, and they swallow it and everything else. Since then, at day 203, we measured activity in tissues that showed in this graph below that. In the red, which are seen the values and increases in the tissues, which range from 13 to 38% depending on the tissues for the 2 different doses and everything else. Then we measured any kind of immune response on the bottom with an ELISA. And you can see that it was a very low immune response to the oral enzyme replacement therapy with the tobacco seeds. We then measured glycogen reduction in the, and we show here at day 203, and the glycogen reduction in the tissues compared to the mock-treated animals range from 18 to 48%, depending upon the tissue, et cetera. This is, long-term stability. This just basically shows that we measured seeds that were stored at room temperature for 15 years and freshly harvested seeds, and we basically saw 10% reduction in enzyme activity. So and I didn't do this on purpose. They just happened to be sitting there in the lab. We then did wondering, what was going on there. We did a simulated stomach experiments, and we found that the stomach environment with pepsin didn't affect the enzyme. The pH is not going to affect the enzyme. The enzyme loves it because it lives in the lysosome. We then tested it with we then readjusted it for the upper GI with the chymotrypsin trypsin, and it didn't affect enzyme activity either. So that the stimulated stomach, which I didn't invent this, other people have done it for other proteins, et cetera. We then went further on this, and we did, we were just doing a dose response and how they respond to different levels of the enzyme. We've initially started treating patients twice a day with 50 milligrams of enzyme, 50 milligrams of seed, I'm sorry. The enzyme activity clearly increased, but then day 24, we stopped. We went to one time a day with 50 milligrams, and it dropped down, as you can see, and at day 41 or 42, whatever it is, we went down to every other day, I believe it is, and then by day, I don't know, whatever day it is, 54, it went back down to the mock treated levels. So we could see that by increasing it, the activity improved. By decreasing it, the activity declined. We did some, and this is all part of the grant and everything else. We did pharmacokinetics with either a lysate or the whole seeds, and as you can see here with the lysate, that the peak levels in the plasma was about 8 hours. With whole seeds, it was about 10 hours. Very simple experiments. We collected material at 0 times, 0, 1, 2, 4, 6, 8, 10, 12, and 24 and 48 hours. Not much sleep during that day. Okay, I'll go through this quickly. This is the urine. We measured the urine excretion, and it was basically the same thing a little bit later. And then we did, the next slide here. We did enzyme kinetics and compared it to Myozyme and placental GAA, and it was very similar or superior in some of the tests and everything else. We did a whole series of experiments. I'm just going to go through, just for the grant and everything else. We measured nicotine levels, which it is none. There was no mutagenesis. There was no endotoxin levels. All of these got to be answered for these kind of grants. And then we're doing a little bit more sophisticated experiments. Really what we think we can relate to more with what's given to the patients with either 50 or 100 milligrams of seeds daily, either ground or whole, for a young and an older group of mice for 1, 2, and 3 months. Preliminary data shows that, of course, that the younger mice responded very quickly, very fast to running wheel activity, grip strength, rotor rod. But the older mice took a longer time to respond and needed higher levels of the DTOB rhGAA. The most surprising thing is that and I mentioned it to Nina, that these animals, even the young and the old, their spontaneous alternative learning by T-maze, after 2 weeks, no matter whether they were young or old, they improved to more normal levels of the T-maze activity in their memory and et cetera and everything else. We did a maximum tolerated dose. We gave it up to 320 milligrams of the seeds, and there was no response there. We did an IND 2015 just to get the response from the FDA, and they gave us some issues and just to see what they were going to think about it. And so we're addressing all of that. Just the last thing, and I'm sorry to delay. Besides this, because it's a tobacco seed, it also can be considered a food source. And it can provide an alternate pathways for an IND and for a pharmaceutical pathway, and that we can be considered a dietary supplement and a nutraceutical, which then you don't have to go through an IND. You can just basically supply it to the patients as a dietary supplement and a nutraceutical, but there's some limitations on how you advertise this stuff, because they don't like to be told specifically for a disease and everything else. And the last slide is just something on propaganda for our company, which is so small. We're doing all these other things. We have it for Fabry's. We're working a little bit on Duchenne's muscular dystrophy. The one thing that we have, which I told Nina last night, is a non-GMO plant product, which is completely different than this, and we're, of course, doing non-viral gene therapy. Thank you very much, and sorry to run late.